Welcome to The Explainer. Today, we're stepping into the high-stakes, completely invisible world of pharmaceutical quality assurance. Think about it. Every time we reach into our medicine cabinets and take a pill, we just assume it's safe, pure, and exactly what the label says, right? But behind that tiny tablet lies an intense, rigorous system of safety measures that most of us never even realize exists. It's this fascinating world where perfection isn't just a goal, it's a heavily documented, absolute requirement. So, how clean is clean enough when making medicine? If you were to walk into a pharmaceutical manufacturing facility right now, you'd probably see gleaming stainless steel floors, workers in full sterile suits, and completely spotless equipment. To the naked eye, it looks perfectly, immaculately clean. But in the world of pharmaceuticals, visual cleanliness, that's kind of just an illusion. We aren't worried about what we can see. We're worried about the hidden, microscopic dangers lurking inside the tiniest crevices of that equipment.
Okay, let's dive into this. The foundation of answering that how clean is clean question comes down to one core concept: validation. Officially, it's the process of establishing documentary evidence that provides a high degree of assurance that a specific process will consistently produce a product meeting predetermined specifications. But in simpler terms, it's basically the ultimate scientific rulebook. It is the act of proving, beyond a shadow of a doubt, that a process works flawlessly every single time. It's not enough for a pharma company to just say a process works. They have to scientifically prove it with hard, documented data.
And what exactly requires this level of absolute proof? Well, under good manufacturing practice, or GMP principles, the answer is pretty much everything. We're talking about the written procedures, the manufacturing processes, the actual equipment being used, the raw materials going into the drugs, the specific human activities, and the overarching computer and facility systems, the whole shebang. Every single one of these variables has to be meticulously tested and proven to achieve expected results consistently. There is literally zero room for guesswork when human health is on the line. Which brings us to our specific focus today: cleaning validation. This is defined as documented evidence with a high degree of assurance that one can consistently clean a system or a piece of equipment to predetermined specifications and acceptable limits. I actually want you to think of quality assurance personnel in these facilities as elite investigators at a microscopic crime scene. Their job is to track down microscopic suspects, these residues you can't even see with the naked eye, and scientifically prove that their equipment is truly, genuinely clean before the very next batch of medicine is manufactured. So, who are these microscopic suspects are constantly hunting? Well, they fall into four main categories. First, cross-contamination from residual active pharmaceutical ingredients, or APIs. Then, ironically enough, the residues from the chemical cleaning agents themselves. Next up is microbial contamination, and finally, any other unintended materials. To make this a bit more relatable, imagine you just used a pan in your kitchen to make a massive, sticky batch of peanut butter. Now you need to use that exact same pan to cook a meal for someone with a severe peanut allergy. You wouldn't just give it a quick rinse, right? No way. You'd scrub it, sanitize it, and inspect it obsessively, because even a microscopic trace could trigger a deadly reaction. That is exactly the level of acceptable limits we're talking about in pharma, just scaled up to massive industrial manufacturing. So the crucial point is that pharmacologically active chemicals may cause clinically significant synergistic interactions. I know that sounds like a mouthful, but it represents the terrifying stakes of this industry. If significant levels of a residual active ingredient from a previous batch, say a highly potent heart medication, are left behind in the manufacturing vat and they contaminate the next product, like an everyday headache pill, those two active chemicals could mix. That cross-contamination could cause an unintended, potentially super dangerous reaction in a patient's body. It is completely intolerable. And this actually brings us to another major hidden danger: the microbial threat. Counterintuitively, routine maintenance, cleaning, and storage conditions can actually provide adventitious microorganisms with the perfect opportunity to proliferate. It sounds a bit backwards, doesn't it? But think about it. The very act of cleaning can inadvertently leave behind pools of moisture. Routine maintenance can introduce lubricants or leave equipment parts temporarily exposed. If a massive stainless steel tank isn't dried perfectly after it's sanitized, it quickly becomes a microscopic breeding ground for dangerous microbes, right there inside the processing equipment. Because the stakes of both chemical and microbial contamination are just staggeringly high, ensuring strict adherence to global regulatory frameworks like the FDA, the ICH, and the WHO is absolutely paramount. They mandate these exact validation standards.
This rigorous tracking and documentation, it isn't just a nice-to-have best practice that looks good on our corporate brochure. It is completely non-negotiable for public safety. Simply put, if a facility cannot prove its equipment is clean according to these specific federal guidelines, they simply cannot make medicine. Let's move to and see how this builds into an actual actionable process. How do these investigators practically prove a machine is clean? Well, they follow 4 distinct validation phases. First up is the planning phase, where they strictly define the scope and select the appropriate cleaning agents. Next is the execution phase, where they actually run the cleaning cycles and carefully collect samples from the equipment. Third is the analytical testing phase, applying highly validated lab methods to test those microscopic samples. And finally, the reporting phase, which involves evaluating all that data against strict acceptance criteria and getting that final quality assurance sign-off. Now, the sheer breathtaking rigor required just to set up one of these tests is massive. Just the first half of a standard 13-point validation protocol is incredibly dense. Before a single drop of cleaner is even applied, you have to meticulously document the specific objective of the operation. You have to define strict responsibilities for exactly who is performing, reviewing, and approving the study. There needs to be a highly detailed equipment description, and you must rigidly track the interval, meaning the exact allowable time between the end of production and the start of the cleaning procedure. Beyond that, you have to define the exact cleaning procedure step by step, specify the precise number of consecutive cleaning cycles, and establish routine monitoring requirements. And wild as it sounds, all of that is just to set the stage for the actual test. Now, what's really fascinating about this next part is the analytical half of the protocol. This is where things get incredibly scientific. Take recovery studies, for example. You actually have to prove that your sampling method, like rubbing a tiny, specialized swab on a massive 500-gallon metal vat, actually collects the microscopic API or detergent residues from that specific surface. You can't just swab it, find nothing, and claim the equipment is clean if the swab itself is faulty at picking up the chemical residue in the first place. Furthermore, you have to document your analytical methods in the lab, establishing the exact limit of detection and limit of quantitation. You must define strict acceptance criteria for residues and detail your product grouping strategy. Every single variable is mathematically and chemically accounted for, leaving absolutely nothing to chance. Which brings us to a final, provocative thought. Next time you take a medication, will you think about the invisible validation keeping you safe? We take so much for granted when we swallow a pill, naturally trusting that it will heal us and certainly not harm us. But behind that quiet trust is an immense, hidden documentary effort, an entire world of dedicated QA investigators, complex recovery studies, and rigid regulatory frameworks. It is a fascinating, invisible shield of science operating in the background, designed solely to guarantee your safety. Thank you for joining me on this explainer to uncover exactly how it all works.